So we're going to continue our discussion of receptors, and we're going to do this lecture. We're going to talk about the next two. So receptors, tyrosine kinases, and receptor guanylyl cyclases. So we're going to start with this one. So receptor tyrosine kinases. So these guys are different from GPCRs because they're a little bit of simpler pathway. They do more of it themselves. So this is a broad sort of cartoon of just how an RTK works. So it generally is a dimer that dimerizes when the ligand binds. So whatever the ligand is, again, there's a whole class. There are a number of different ones that are possible. But when the ligand binds, it's going to bring the dimer together. And so it's got two domains. There's an extracellular domain of each one, and then there's an intracellular domain. So this is outside, of course, because that's where the ligand is, and this is inside. So no. And so what happens on the inside when the ligand brings these two together is these interior domains actually phosphorylate each other. So to the right, they're called receptor tyrosine kinases. The receptor itself is a kinase. And so this domain can phosphorylate this one, and this domain can phosphorylate this one. So that's going to happen after the ligand binds and it dimerizes and it brings it together. Then at that point, usually what happens is these now phosphorylated sections of the internal domain are going to cause another protein to come over here, bind, possibly get phosphorylated, and lead to further intracellular signaling. RTKs do not usually use second messengers. They're generally going to instead keep activating proteins and instead of creating a small molecule. Also, in the whole pathways for RTKs, we're just going to see tons of kinases. So a lot of things are going to be kinases. One example that we will use is the insulin receptor protein. So the insulin is the signal here. Insulin is generally speaking, a signal of high blood sugar. So after you eat a meal, your insulin is going to rise. And so all the downstream effects are going to be dealing with that, the fact that you just provided your body with energy. So if we have here the two, in the light pink and then the dark pink are the two subunits that are going to come together in the dimer. And here this is showing just that when these two internal domains come together, again, it's going to phosphorylate each other. This is only showing one of them being phosphorylated, I think, to compare, but they would both get phosphorylated by each other. In this case, it gets phosphorylated 3 times, and that causes a dramatic shift in the substrate binding site. So when these 3 tyrosines are not phosphorylated, they are going to be sort of covering up the active site. But when they get phosphorylated, they kind of move away, and then other proteins can come in and get phosphorylated by this kinase. So the main one we're going to talk about here is going to be IRS1. IRS1 is going to come in and get phosphorylated by the activated insulin receptor. And so we can see here it's got 2 phosphates on it, or sorry, 3. It's got 3 phosphates on it. And once that happens, that is going to cause the coming together of a rather large conglomeration of proteins. All right, so we've got this GRB2, SOS, RAS, and RAF1. RAS, we can actually see here, is a G protein. It is going to cleave GTP to GDP. And that is actually, so this is a G protein. It's not a G protein that is linked to a receptor, so it's not part of a G protein coupled receptor, but it is considered still a G protein. The part of GRB2 that binds to this phosphorylated section is called an SH2 domain, and this is another example of modularity where we have a lot of different domains called SH2 domains, and they generally, when you see an SH2 domain, it's going to be binding to a phosphorylated residue. So there's a domain of this protein that's kind of its purpose is to bind there. So all of this is basically to activate this protein right here, RAF1. So RAF1 is a kinase. So also, I'll just point out again, right? We haven't created any, there's no small molecules going on here, no second messengers. RAF1 is a kinase. RAF1 is going to phosphorylate this protein called MEK. MEK is also a kinase. MEK is going to phosphorylate this protein called ERK only when it is phosphorylated, right? So it has to be activated. So it's going to get phosphorylated and activate ERK. ERK then is going to actually move into the nucleus, where it is also a kinase, and it is going to phosphorylate some transcription factors. So transcription factors are just anything that causes a specific portion of the genome to get transcribed into mRNA, and then which will eventually get turned into protein. And so there's a lot of transcription factors. These particular ones are going to activate genes that are needed for cell division. So if you have a lot of energy around because you just ate food, you might imagine that's a good time for your body to be doing cell division because it needs energy. So that's why this portion of the pathway is going to go down that route. This is not the only thing that is going to happen, but that's why this portion is happening there. So this RAF1 MEK ERK situation, where RAF1 activates MEK, which activates ERK, which is a kinase itself, is an example of something called a MAPK cascade. And the reason for that is this is actually pretty common, where we have series of kinases. And the MAP stands for mitogen activating protein. So the MAPK is the one that is going to activate the transcription factor. So ERK would be the MAPK, but then you just add an extra K on. So ERK is a MAPK, MEK is a MAPKK, and RAF is a MAPKKK. They really did not think this through when they were naming it. Apologies for that, but right. So it's a MAPK kinase. Sorry, it's a MAPK kinase. Yes, the kinases get kind of out of control. So it's a mitogen activating protein, kinase. So it can kind of help you keep track of what's going on. And this is just one example of a MAPK cascade. There are other examples in different signaling pathways. So insulin signaling is going to do more than just lead to the activation of some genes for cell division, because there's a lot more that needs to happen when you have plenty of energy around. So this is just a different branch. So here's IRS1 again. So we're just going to start from that. So IRS1, the previous thing started that whole conglomeration that started the MAPK cascade. Another thing it can do is it can come over here and instead bind to this PI3K, which it also has an SH2 domain. Remember, the thing it bound to in the other part of the pathway was also had an SH2 domain, so that's, again, modularity. And that is going to convert this PIP2 lipid in the membrane to PIP3. This is one of those inositol lipids. Don't get confused. We had an IP3 last time. That was just the head group. This is still a lipid. It has even more and more phosphate on it than previously. But it's still a lipid. It's still in the membrane. So this PIP3 is going to bind to and activate PKB, just our protein kinase B. And this can do a couple different things. I'm just going to jump down here, actually, because I think this is a little bit the simpler part. One thing it can do is it can come over and it can stimulate GLUT4. So the way it stimulates is it causes GLUT4 to get moved from internal vesicles into the membrane. GLUT4 should sound sort of familiar. We've seen GLUT1 and we've seen other glutes, but GLUT4 is just a glucose. It's a passive glucose transporter, so it's going to allow glucose to come in from presumably the bloodstream out here, and so that is going to increase the uptake of glucose because glucose is high when we have insulin around. And you have to take the glucose up before you can use it. So then we have the other half of this, and what this is going to do, so we have this GSK3, and this is glycogen synthase kinase 3. So actually, let's pause on what that does for a second. Over here, we have glycogen synthase. Glycogen synthase is exactly what it sounds like. It is the enzyme that catalyzes the synthesis of glycogen, which is what we use to store extra glucose. So this is something we would want to be happening when we have high insulin. So we want our glycogen synthase to be active. And what activates, so this GSK3 is actually a protein that when active is going to inactivate GS. So we don't want this to happen when we have high insulin. Right? So actually this is the default. This is what's happening before the insulin pathway started. And when the insulin pathway goes through this IRS1, PI3K, PKB situation, what PKB is going to do is put a phosphate onto GSK3, but I think for the first time, phosphorylating GSK3 inactivates it. All right, so it's going to inactivate GSK3, so we inactivated an inactivator, so that's going to make GS more active. So because GS is also something that when it gets phosphorylated, it is inactive. So it can go either way. I think many times we see, I think probably more often, we see proteins that are only active when phosphorylated, but it is also perfectly possible for proteins to only be active when they are not phosphorylated. So throughout all of that pathway, the gist of this is that when the insulin pathway is turned on, we get more active glycogen synthase, and we turn on synthesis of glycogen. And we also turned on the bringing in the glucose because we're going to need that to make our glycogen. All right, another RTK, again, just so we don't get the idea this is the only one, is called the EPO receptor. And this is also sometimes called the JAK-STAT pathway. It's the same thing. So EPO is a shorthand for erythropoietin, which is just a small protein. And so again, the protein is going to bind in here, and it's going to bring together these two dimers, or these two portions to make a dimer. And when that happens, and they phosphorylate each other, it is then going to recruit two proteins called JAK and STAT to it. They sort of skip that portion. JAK and STAT then, when they come over here, STAT's going to end up getting phosphorylated. So STAT actually, again, has an SH2 domain, which is binding to phosphorylated residues. So it only comes because it can bind to this EPO receptor phosphorylated residue, and then JAK is a kinase that phosphorylates STAT right here. Right, so, and then once STAT is phosphorylated, it's got this SH2 domain that likes to bind phosphorylated residues, and it actually dimerizes with itself by binding the phosphorylated residue of the other molecule. And then STAT, as a dimer, goes into the nucleus, and it is actually a transcription factor itself that is going to cause the gene expression, and in this case, of genes that lead to red blood cell maturation. So you may have heard of EPO before. I don't think this is as common anymore, but in maybe the 90s or 2000s, this was a really bit popular doping technique for especially people like cyclists. So people, I honestly don't remember what it was Lance Armstrong was taking, but people that were, wanted to be on the professional cycling tracks and things like that, would take EPO, and the reason for that is because they would get more red blood cells out of it, and more red blood cells leads to more oxygen being able to be brought throughout your body, and that gives you a better ability to do cardiovascular activities such as long-distance cycling. So it's obviously banned because it's doping, so people generally don't do that anymore, or if they do, they're finding ways to mask it. Obviously, unfortunately, doping is still a very real problem in many sports, including cycling, but that is why they were doing it. So just another side note, there's lots of different pathways, again, with this sort of modularity thing. There's another portion of this pathway where a different protein can combine here, and then GRB2, which we actually already saw. We saw GRB2 in the insulin receptor pathway. GRB2 can now lead to a MAPK cascade that goes and does the same thing. It still goes and affects gene transcription. So different things that can happen there. So because we keep seeing things in sort of various different situations, we actually can start to see crossing over of pathways. All right, and so one thing that you might think might cross over is the insulin pathway and the beta-adrenergic receptor pathway. And the reason for that is the beta-adrenergic receptor pathway was trying to get you to get glucose. The insulin pathway is trying to get you to store it. And I actually don't want to stress too much about this because I think it gets a little complicated, but I think the point I want you to know is that they can interact with each other. So in fact, actually the insulin receptor can phosphorylate to the beta-adrenergic receptor, and the beta-adrenergic receptor can also actually affect the insulin pathway. So things can be very related, especially we keep seeing these same things like PKB or GRB. We keep kind of seeing them come back. So this is going to be, everything's connected. That's kind of the gist I want you to get from here. I don't want you to worry about the nitty-gritty details like you would for the pathways themselves, but everything's connected. All right, the third class of receptors here is the receptor guanylyl cyclase. And this is, again, going to be a little bit more simpler one, kind of like a little bit more like the RTKs than the GPCRs. So the example we're going to use here is the ANF receptor. ANF stands for atrial natriuretic factor, which is just a peptide released when the heart has blood volume. And so what we want to do is actually increase salt secretion and change the blood volume through osmotic changes. This is, I think, a lot less clear what the downstream effect is, so I don't want you to worry too much about that. But you should definitely know what it does. So receptor guanylyl cyclase is going to mean it's going to cyclize guanosine monophosphate. So we saw an adenylyl cyclase in the epinephrine pathway. This is just a guanylyl cyclase. So instead of taking ATP and going to a CAMP, it's going to take GTP and go to CGMP in exactly the same way. This I kind of circled it stupidly, but this OH comes and attacks the phosphate and cleaves off the pyrophosphate. All right, so again, this is like the RTK in that it's a dimer that comes together when it binds the substrate, or when it binds its signal. And when it comes together, then the catalytic domains are activated, and it's going to cyclize this CGMP. CGMP can then go and be a downstream. This is going to be a second messenger, of course, and to do a whole bunch of different things, apparently including in the heart, affecting the osmotic pressure. So there's actually a subcategory of the guanylyl cyclases, and these are unusually cytosolic. So cytosolic obviously means it's in the cytosol. It's a soluble protein. It is not in the membrane. And that would be a problem for most receptors because their signals are usually not able to pass through the membrane. So they have to be on the outside. However, this one's unusual in that it's activated by nitrogen monoxide. And so this guy is small and relatively nonpolar and can pass on right through. This still performs the same reaction. It's still going to cyclize GTP into CGMP, but it just does it cytosolically. So this is the main thing to know about this, that it's unusual. It is in the cytosol. All right, and so that is those two sections, and we will finish this up with the next lecture.